Hi, grade 10s. So today we're going to continue our discussion about how substances enter or leave cell membranes. So last time we talked about passive transport and the different types of passive transport, and those are the following: osmosis, diffusion, and facilitated diffusion. And those all involve going from a high concentration to a low concentration, aka down the concentration gradient. Well, today we're going to actually talk about something called active transport. So the last one I said was passive, this one is active, which means still talking about transportation. And why is it called active? Well, that is because energy is actually required to move material up the concentration gradient. So in this case, we're actually going, from a low concentration to a high concentration. And that is why it's going up the concentration gradient, because essentially it's going against the flow. It's not what would occur naturally. And in order for that to happen, because it's not going to just allow that natural flow from high to low, instead it's low to high, it actually requires energy. And that energy is in the form of what we call ATP, which you get into further detail and conversation about in Bio 20. Okay. So active transport is often used, in regards to large or charged particles. And in order for active transport to happen, you often need that protein involvement again. Okay, so if you were to watch this actual little clip here, you will see, your cell membrane, you see a high concentration of glucose on the bottom, low concentration on the right, ATP right there, it gets energy, and the glucose is going to move from low concentration to high concentration. So therefore, that is what we call active transport. Okay? So transports larger charged particles, like I said, it needs ATP, it needs energy, and we often require a carrier protein. Okay? So we are going to look at different types of active transport. So we have endocytosis and we have exocytosis. And then there's actually different types of endocytosis. So the first one we're going to look at, is endocytosis. Endocytosis is what occurs when substances, endo, that means they are entering into the cell. So the cell membrane basically opens up, it forms a pocket around the material outside the cell that's floating out there, and it folds in, kind of captures it, creates a little vesicle, and then it can enter into the cell membrane. So the two different types of endocytosis are phagocytosis and pinocytosis. So phagocytosis is what I think about when I think about it's eating. And the reason I say eat is because it is taking in large particles, or other cells. You can see in the diagram below here, it actually kind of looks, that's the red blood cell. So it's essentially eating it, it's taking it in. So endocytosis. Now, pinocytosis is still taking it in, but this is actually more concentrated on fluid. I think Pinot Grigio is actually a form of wine. So I think Pinot in regards to wine, aka drinking, that's a fluid intake. So in this diagram, solute, that's your fluid, that cell membrane is opening up a pocket. What's happening is it's forming and enclosing it. And once it's enclosed, it has this vesicle of liquid inside of it that is now available for the cell to use. So exocytosis. So this is exo, meaning things are exiting the cell. Same process, except instead of taking into the cell, it's going out. The cell membrane is going to form a pocket around material that needs to leave the cell. And once it forms that pocket around it, it basically goes into the membrane and opens up, and it's released outside of the cell. And I'm going to let you just watch this little recap on endocytosis and exocytosis.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Okay, so that's it for active transport. So a little quick highlight: active transport in general is from low concentration to high concentration, so going up the concentration gradient. And we have different types. We have endocytosis and we have exocytosis. So endocytosis is the taking in, exocytosis is the substances leaving. I hope that makes sense. You'll have a couple kind of questions on it, and have a good rest of your day. Bye, guys.